Here's a live run through of an AS Biology paper 2. So, first question, what is a gene? Very simple. Section of DNA that codes for a polypeptide, not a protein, a polypeptide. That's what you need to know for AS level. Next, about the production of messenger RNA in a eukaryotic cell. How is that different to producing it in a prokaryotic cell? So remember, this is about production, not the location of transcription, for example. So try not to fall into that trap mentioning about the nucleus. However, it's good to mention anything you can, as long as it's in a coherent manner and they don't conflict with each other. So this student did mention about nucleus, but later on he did say that eukaryotic DNA undergoes RNA splicing as it contains introns, which prokaryotic DNA does not contain, and therefore that does not happen, which is 2 marks right there. The idea about no introns and RNA splicing, only in one. Next is about a short single-stranded artificial nucleic acid called PNA binding to a small section of DNA, how that affects the transcription of that section of DNA. So here, this is kind of like a bit of enzyme theory, similar, protein structure theory. So you have to mention the idea of complementary and tertiary. Those are 2 key words that come with almost all of these questions. So right here, PNA has a complementary tertiary structure to the section of DNA, so it will bind to a section of DNA. Inhibiting or preventing RNA polymerase enzyme. It's good if you know the specific enzymes, it'll just help you get more marks. But mentioning an enzyme in general should get you the mark in general anyway. But it's good to be specific. Enzyme from binding, therefore it will reduce transcription. So either of those 2, that's 2 marks. Easy. Next, the role of ATP in the process of translation and protein synthesis. Very simple. However, once again, there is keto. First of all, the first mark, which is always an easy mark, don't drop these easy marks, is that ATP releases energy. Define what ATP's role is, first of all, its function, what it does. Why is that important? Well, that's going to be important in one of 2 things you can mention. First of all, you can mention the formation of peptide bonds between adjacent amino acids. That's normal, as it is. The second one you can mention is the attachment, releasing energy for amino acids to attach onto tRNA. So attaching onto tRNA is the key, or joining onto tRNA, because that says it kind of explains the concept of it, rather than saying just bringing in the amino acid or getting the next amino acid in the line. Be specific. It's joining to the tRNA molecule, which is involved in the translation process. Next, about the term used to describe different structures. Alpha glucose, beta glucose, that's fine. Just isomers, very simple. Especially if it's a structure, shouldn't be too difficult. Next is about a colorimeter. Colorimeter is simply a device that measures the rate of absorption by passing light through. So right here, just make sure you're specific and you use quantifying words like higher or lower. So a higher absorption will indicate a higher sugar content. Easy mark. 2 control variables, the volume of solution or temperature of solution. There's other ones as well, like pressure, for example. But as long as you have 2 locked in, that's it. Okay, next, calculation. So, very simple maths here, 180 times 5 over 6 to get the flesh tissue, the mass, and then you times that by 0.08, which is the percentage of the apple that is sugar content, to give you 12 grams. Now, this is a little tricky, but the first thing to notice is the idea of water potential. And actually, before that is the stain going from black to less black after being stored for a week, the apples. Water potential, apple juice decreases when apples are stored. Why does it decrease when apples are stored? Okay, so less black. What does that mean? So black is a test for starch. Therefore, less black means there is less starch. So, from that we can infer that starch is hydrolyzed. Use that terminology. More starch is hydrolyzed when it's stored for a week. And the reason this is important is because starch is insoluble. That's a property of starch. That will get you the mark. Starch is insoluble. Or reducing sugars, specifically maltose in this case, which is difficult to always get, but once again, just like with the enzymes before, it's better if you can mention the exact name. So maltose, as a reducing sugar, will be in a higher concentration. So either way, whatever you say, starch is insoluble, it'll just mean that there will be an increase in solute concentration within the solution, because there will be reducing sugars in the solution, which will then reduce the water potential. Decrease when apples are stored, which essentially means that in that case scenario, water will have a greater tendency to move into that area. Now, and that's just the nature of apples. Two structures, increase the rate of absorption by the cell. Once again, more specific terminology here. Increase surface area for diffusion or facilitated diffusion or active transport. Those kind of keywords for a type of transport, not just for absorption. That'll be 1 mark. And high blood circulation is what the student put here, which is not a bad idea. However, it's not as specific to the question as it requires, because they want you to explain 2 ways that the structures shown increase the rate of absorption. So identifying many mitochondria in the cell will help you to see that this releases energy for active transport. Once again, that specific process. Next is about simple calculation question. 2 times 3.14 times 0.05, just knowing how to play with radius lengths, that's fine. And timesing it by 1,000, the student did lose out on a mark here because he got the final area in micrometers squared, but he misread the part where it said 1,000 microvilli. Remember that. Use the parts of the question to your advantage. And from that, you will just add the existing surface area. Don't forget that the existing surface area already, and get the ratio of that. It will just give you 41 to 1. Next is about protein Z01 found in the surface area of ileum cells. Anti-Z01 monoclonal antibody to identify cells in the sample. Okay, so this is just basics of monoclonal antibody. There's very logical structure for 3 of these marks. 1 mark that you can also say is about Z01 being located on a cell surface membrane, but the main 3 marks that you're likely to get if you know how to apply your knowledge of biology is the monoclonal antibody has complementary tertiary structure, or complementary to the Z01 protein, so it will bind to the Z01 protein, with the monoclonal antibody having a fluorescent dye attached to it that will be released on binding to that protein, which can then be seen on the optical microscope, of course. 1, 2, 3 marks. Next is about the process of meiosis. Results in haploid cells. This is going to be very simple. You just have to talk about the idea of there being 46 chromosomes, originally diploid cells, which DNA will be replicated, homologous pairs will be split. Just talk about the process of meiosis. Homologous pairs of chromosomes are split, and then the sister chromatids are split, which will then go from a diploid to, again, diploid to a haploid, because there's 2 divisions. That's why it produces haploid cells. Okay, next is more about molecule-independent calculate how many cells. That's not too difficult at all. Just use the segregation. Identical arrangement. Right, are expected to have. Okay, diagram. Show closure will produce meiosis in 1 gamete. So you'll just show them as the 23 single chromatid-like chromosomes. Here, this is a process of mitosis. Don't be fooled by the fact that there are 2 divisions here. The student did put meiosis here, but it is indeed mitosis. Why? Because it says diploid spores, and in the question it also says a mature zygote. So this is after fertilization, so therefore it has 2 haploid cells, a diploid cell, dividing by mitosis. Grouped in a phylum. Okay, very simple. Just talking about the kingdom, phylum, class, order. All those things based on their biomechanics, you can say. Biomechanics, whether they breed, environment, all these things. Features, genetics, all that. Any of those will get you the 2 marks. Scientific name, you just get the genus and the species. So it will be, yeah. Taxon will be, that's the species, that's the genus, that's the family, and that will be order. It'll be the order. Okay. Fine. Likely phylogeny between these species. Percentage difference. You just compare the 2. You know, white is closer to black. with the reference Indian rhinoceros. So white to black is fairly far, but they are close to each other. Indian is close, you know, those kind of things. Explanation. Okay. Now, risks. Sheep's heart. Okay. So here, cutting away from you is always going to be one. That's also in chemistry, so bear that in mind. Cutting away from your body. Pointing it downwards or protecting it is a better way to say it. Protecting it while moving around, protecting the blade. And also, this is the final mark the student didn't get. Cut on a hard surface. This almost always comes up, and people get confused as to why that is. Well, if you cut on a hard surface, it's less likely to slip and less likely to inflict harm. That's all. Next, application. Remember, paper 2 is a lot about application. So even if you don't know all the content, you know, and you're practicing for an exam that only has specific topics on it, or specific multiple topics, but 1 and 2, 2 and 3, anything like that, it's still useful to get the practice in, unless it's like completely outside your expertise. Like, you don't need to be answering those questions at all. If it's meiosis and you're only doing topic 1 or something. But other than that, you know, attempt every question. It's always useful. Calculate total volume of blood pumped. So here, this is once again application, and this is a little tricky of a question, but as long as you use your brain, it's not too difficult. You see 120 is going to be as high as it goes, meaning this much will be pumped out, which is a total of 82, and 0.8 seconds for 1 heartbeat. So 82 times 60 over 0.08, because they want it for 1 minute, equals 6,150. Pre-high BP, high BP. Now, this is quite a tough question, and there's a different amount of marks, but first of all, recognize pre-high BP and high BP. Here, the student did do very well. He did get 4 out of 4. However, he did miss some easier marking points, you could say. So, first of all, stating that treatment 1 reduced it by minus 1.7, treatment 2 minus 4. That's always important. So, treatment 2 reduced it by more, reduced blood pressure more. And you can even say pre-high. So, however, in the mark scheme, though, they want you to say that pre-high BP is still abnormal. So it's good to mention that, because pre-high BP is nowhere near normal yet, still above average, or even the much more logical explanation that treatment 1 still remains in a very high blood pressure, so it doesn't reduce the risk at all. So those are 2 or even 3 easy marks there. We'll say 2 there, if you get one of those points. Other points are like here, which is side effects. Treatment 1, as long as you use the data, it's okay. 3 blood pressure medicines. Treatment 2. Treatment 2 will have unknown side effects that we don't know about. And as well as that, duration. So in terms of like testing, you need to know these like easy marks you can pick up. So we don't know the duration of the trial. And we do know that there is a large sample size here. So this easy mark is actually far simpler to integrate now, because we can simply say that the large sample size shows that it is representative, it is realistic. And yeah, there's also a mark for neither treatment actually brings it back to normal BP levels. But as long as you say 2 is more than 1, and both are not ideal, get these key points, and then some easy ones about like large sample size, duration, all those things. Okay, next. Very simple. They just want you to, so remember to read the question. Just the arrangement of tissues. I've had students that have drawn individual cells and taken very long, and that's not what they're looking for. And you can correct it, in fact, if you mess it up. Right here, I'll actually show you that the students' work. So this was done with the 4 out of 4 here. They did go into circles a bit for this question, but they did get the final points there. Final key points integrated. And for this one, they did manage to save it, showing the different layers. So they show clearly the upper epidermis, palisade, spongy mesophyll, and lower epidermis there. Wax cuticle as well. And they labeled palisade mesophyll. So very easy 3 marks. Very easy 3 marks. After this, standard deviations. So this is topic 3 and 4. When you're dealing with these kind of questions, always use the key terminology, always use the fact that we're dealing with standard deviations. Standard deviations show statistical significance by measuring the plus or minus, like the uncertainty around a certain value. And if this overlaps with another value, that means it is statistically insignificant if you're trying to find a change, or if you're trying to find a correlation, it's statistically significant. So make sure you remember that. And the student did get this perfectly with 4 marks. What can you conclude about the effect of different components on the shelf life? Well, mean number of stomata, the standard deviations overlap, so it's not definitive as an effect of the mean shelf life. So remember to read the question, they want the effect. That's their mean transpiration rate. Standard deviations do not overlap, so this is significant. And then explain on top of that, this was because higher temperatures increased the rate of water loss due to more kinetic energy in molecules, so to retain water, less transpiration. That's fine. No, didn't actually get the mark for the explanation. It's just talking about significance here. This is about explaining the effect. Now, data shows that the SDs don't overlap, so there is an evidence of an increased shelf life, right there. Significant. And yeah, it's 4 marks already. Greater volume of water used in processes. And finally, other than a change in temperature, give one change a student can make to the environmental conditions to increase the shelf life. Well, slightly wrong trajectory here about increasing light intensity, because reducing light intensity is more important, and you've got to see the effect of why. Reducing light intensity will lead to more stomata being closed, decreasing the rate of transpiration, and as we've seen, this is linked to shelf life. It will increase shelf life. Or increasing humidity is always a classic one, lowering the water potential gradient as there's humidity near, which will lower the rate of transpiration, lowering water loss, increasing shelf life. Because high humidity, it means that water is less likely to actually move out of cells but into the air, out of leaves. From there, we can carry on. And Guppy, this is just one of the things you have to know. Null hypothesis. So null hypothesis is always about nothing is happening. So as the generations continue, there is no increase in the mean number of spots per fish. That's the null hypothesis. There is no detectable change of any sort. Further investigation, the student can confirm. Over a longer duration, larger sample size, anything here works. Further investigation. Had affected guppy behavior. You could do a different type of guppy, species of guppy, different ages. Anything here would work. Type of selection. Number of spots per fish. Predators added. Okay, predator added, mean number of spots decreased. So this is directional selection because it's going to a specific end. It's not going to a specific value, and it's going to a specific side, which is the lower number of spots of fish. Directional selection. And explain on top of that why this environmental change will reduce the number of spots per fish because it makes them easier to see, the spots per fish. Always explain why that feature is disadvantageous, which means that ones with less spots have a higher likelihood to survive and reproduce, passing on their favorable alleles to their offspring. And structure of insect gas exchange system, you just need to know about the little folds they have, so as they move they release gas. This is just ideas you have to know. Provide cells with sufficient oxygen, diffusion processes as well, high surface area, all those things, limits water loss, movement also for gas exchange, as I said, and limits water loss through cells, different systems. Not too difficult. How humans breathe in and out. Well, diaphragm contracts, thoracic cavity expands, allowing air to come in. Why? Because as the thoracic cavity expands, lowers the pressure. So there's a pressure gradient actually from the outside air into your lungs, which allows air to go through. But this pressure gradient is smaller than you think. Because when you breathe normally, I'm breathing normally, there's a very small tidal volume that I fluctuate between. If I breathe consciously, I'm using energy, I'm using my sympathetic nervous system, so I'm actually using energy for that. It's a bit of like active transport there, so that's when you get the big breath in. You need active knowledge of that. If you're breathing through a parasympathetic nervous system unconsciously, there's a small tidal volume because the gradient is actually not that big, so when you're unconsciously just breathing in and out, equilibrium is reached quite quickly. Breathe in, air comes in, then breathe out, carbon dioxide removed, thoracic cavity goes down, diaphragm relaxes. Alright, got some muscles. And that is it. That is a live run through of AS Biology Paper 2. See, not too difficult. easily get this. This student did achieve an A star as well, which is very good for him. And hope you learned a lot. Stay tuned for more videos.